Hello. My name is George. I'm a fourth year biomedical science student at the University of Ottawa. I'm the president of the 2020 cGEM uOttawa team, and I'm a previous gold medalist with the 2019 cGEM uOttawa team. Now, this year, I've been called upon to provide a mentoring and guidance role for the uOttawa cGEM team, and on behalf of the entire team, who have been working tirelessly all summer on their respective projects, I will be presenting the work of uOttawa's 2020 cGEM project. So before we get to the team, I'd like to give a quick acknowledgement to Dr. Mads Kern, a longtime champion of cGEM and biotechnology initiatives at the University of Ottawa. Right at the start of the summer, in the light of the COVID-19 pandemic, we recognized that a lot of research initiatives were shutting down and students were losing jobs. So Dr. Kern and I, we wrote an application, we applied for some funding, and fortunately, we created the BioGenie program at uOttawa in partnership with BioTalent Canada and the co-op office at the University of Ottawa, where we secured 50 fully funded co-op positions for students working in biotechnology-related projects, and a list of projects can be found here. And so the cGEM project mainly revolves around the operations of what we called the BioEnvo team. And that brings us to our lovely team members. So Hillary is a representative of the microplastic filtration subteam, which focused on designing a clarifier insert to go into wastewater treatment tanks. Vanessa and Jennifer are representatives from the microplastic remediation subteam, who focused on how to process the collected microplastics and prepare them for biodegradation or recycling into new products. Ricardo is the representative from the microplastic biodegradation subteam, which looked at optimizing enzymatic processes to degrade microplastics. Hertek and Mohammed were involved in other BioGenie programs, but they also served as the cGEM representatives for uOttawa. And finally, Kirsten and myself, we were here to support the team and help bring all the pieces together. With a good mix of science and engineering students, it was a very collaborative effort this summer. We met frequently, online of course, about twice a week to discuss research updates and to share with each other our newfound areas of expertise. It was also a chance for team members to ask each other questions and to clarify details with other sub-teams and other members. And of course, with different educational backgrounds, some concepts needed to be explained more thoroughly to the group than others in order to ensure that everyone understands the research, the decisions being made, and the continued development of the project. We did find that our team was the most efficient when everyone was on board, when everyone was aware of the research that others were doing. It really allowed all three teams, sub-teams, to assist each other and to work together towards our common project goals. Now, it's worth noting that our project is almost completely theoretical, so we did not actually gain access to the lab at all this summer, and as such, most of our work is essentially a proposal based on extensive literature review. So we hope that while you listen to our project, you can understand and appreciate some of the limitations that we face, but we hope you can appreciate some of the science and the research nonetheless. So, the focus of our project was to find a biology-based solution to collecting and remediating plastic-polluted freshwater environments. Now, it's universally accepted that plastic pollution poses an imminent threat to wildlife, ecosystems, and communities across the globe. This microplastic problem has actually become a pressing environmental concern, right? Leading the World Health Organization to call for more research into microplastics and to crack down on plastic pollution as of August 2019. And global plastic production was estimated to reach 322 million metric tons in 2015 alone, with future projections anticipating an increase in both production and use over time. So in other words, plastic pollution is an ongoing and intensifying problem, and we really need to take action to prevent further pollution of our aquatic ecosystems. One source in particular that we looked at was the treatment process or imperfect treatment process that occurs at wastewater treatment plants. So our team proposed a novel start-to-finish procedure that could be implemented in wastewater treatment plants to reduce the release of microplastics into the environment. First, we proposed the implementation of a clarifier, which could capture and separate certain types of microplastics. We would treat the PET plastics, polyethylene terephthalate microplastics, through enzymatic depolymerization using PETase enzyme from I. sakaiensis, which has been shown to degrade PET microplastic. To facilitate that reaction, we would implement a bioreactor utilizing a fed batch culture technique to bring together the collected PET microplastics and the PET-A's enzymes. The enzyme would break down the polymers into its monomers, which could be utilized to make recycled plastic products. The various other non-PET mixed plastics could be collected and processed into biogas for combustion or pyrolysis. Overall, this process allows for remediation of microplastic polluted waters and allows for recycling to occur. In terms of modeling, we produced a very basic mathematical models to predict the efficacy of our devices, and we determined that our clarifier should remove approximately 93% of microplastics from the influent or liquid flowing into our system. We would also hope to track the efficacy of our mutant enzymes with Michaelis-Menten kinetics and equations, but of course, with a lack of data and concrete numbers, there wasn't much that we could do in this regard. But for experimental proposals, there were three main studies that we could conduct if we had the opportunity. So the first experiment would be to recreate the result of Fionn Ferreira's ferrofluid experiment, which used magnetite powder in water containing microplastics to create a ferrofluid, or a substance that can easily be removed from solution while carrying the plastic with it. Now, this technique was found to remove 87.6% of microplastics, and as such, our team wanted to extend that proof of concept to primary clarifying tanks in wastewater treatment plants to collect microplastics on a magnetized attachment that we designed ourselves. So we hypothesized that ferrofluid liquids would form as it did in the original experiment, assuming that other microbes or debris that exist in the primary clarifier do not impact the effect of the magnetite powder. We would test our magnetic clarifier in the lab with various influent liquids, so plain water, water containing microplastics, and water from the Ottawa River, to evaluate the efficacy of our clarifier. We could also compare it to existing industry tools to see if our product makes a significant improvement. The next thing we could look at are the various mutated PET-A's enzymes. This would be the standard cloning, transformation, and protein harvest procedure. We would clone the PET-A's gene onto a plasmid with a sec-dependent signal peptide and a his tag, then transform it into a strain of E. coli. The team mentions K12 here. It has been verified that the sec-dependent secretion pathway used in I. sacchaensis to translocate peptase enzyme is compatible in E. coli, and hence the specific design of our plasmid. Now, after growing the cells, lysing, centrifuging, collecting the enzyme through metal ion chromatography, we can verify its presence through Western blot and its function through enzymatic assays with PET plastic substrates. Now, the team also explored the induction of various point mutations that have been shown to increase both thermal stability and enzyme activity. An experiment involving the aforementioned steps before, just previously, could be designed to determine which mutant is most effective amongst the other mutants and compared to the wild type enzyme. Of course, we would use Michaelis-Menten kinetics to monitor such activity. Some experimentation will also be needed to determine optimal operating conditions for our novel bioreactor as a reaction site for our enzyme. Based on existing scientific literature, our proposal aims to use a fed batch system with a solution pH of 9, glycine sodium hydroxide buffer, a heating jacket to keep the temperature at 40 degrees Celsius, and a stirring mechanism. And of course, any of these variables can be modified in independent trials to determine the optimal reaction conditions. Finally, for pyrolysis experiments, previous literature shows that maximum degradation of microplastics occurs between 420 to 490 degrees Celsius. We would test the degradation of these plastics in a miniature pyrolysis chamber at varying temperatures between 420 and 490 degrees, with and without a catalyst, so either zeolite or palladium, to determine the optimal plastic decomposition conditions. And that pretty much sums up the experiments. Now, in terms of science communication, we did the usual social media shoutouts. We had our members discussing their projects and activities on their own individual social media pages. What was super exciting is that we organized this big conference and virtual celebration at the end of the BioGenie program with over 200 participants across 4 different sessions. We invited each BioGenie team to come and present their work, around a 20-minute presentation, to other uOttawa students, industry leaders, and uOttawa faculty. And that was a fantastic experience. A lot of good science being shared, lots of meaningful connections made, and a great segue into this year's cGEM presentation. Now, for human practices, to inform and refine our research design, we consulted a variety of industry experts to seek guidance, feedback, and potential partnerships. We reached out to companies in mining, pyrolysis, and char industries. They provided valuable insight on current market trends and existing products. For example, they mentioned that the most profitable product from pyrolysis for our purposes would likely be liquid oil, which is why we focused on producing that with our system. For char, we were told that it was actually not possible to compete with the soil and fertilizer industry, as biochar simply just costs too much relative to commercial fertilizer. And many of these companies also offered us a chance to test some of their machines and equipment, and a couple of pyrolysis companies expressed that they would be more than happy to work with us in the future if we were to get some more practical data. So why is this all important, right? Well, microplastics pose a potential risk to the health of humans and aquatic species. Microplastics affect water quality and indirectly the health of ecosystems and organisms, and they're also capable of absorbing other pollutants and toxins. So these plastics can be formulated to contain lipophilic chemical additives, which can bioaccumulate in tissues and disrupt endocrine functioning and reduce fertility. Ultimately, our proposed process for enhancing microplastic collection and remediation aims to decrease the number of microplastics that can bioaccumulate throughout the food chain, which has both direct and indirect benefits for human health. And just to give you a quick numerical sense of the impact that we can make, based on a local wastewater treatment plant that processes an average of 545 million liters of water each day, if we assume inflowing water contains about 15.7 microplastics per liter, which is a value supported by the literature, this would suggest 8.56 billion microplastic fragments entering wastewater treatment plants each day. Now, if we assume the 87.6% efficiency based on previous ferrofluid data and the model of our clarifier, we would successfully remove 4.15 billion microplastic fragments each day. That's a lot of plastic that would be taken out of the water. In terms of integrating industry feedback, a big thing that came out of our conversations was to target industries rather than consumers. So industries produce the majority of the waste, and it's easier to target industry as they're more concentrated and they're required to uphold certain requirements. This influenced our proposal of collecting microplastics from industrial sources, such as wastewater treatment plants and industrial washing facilities, such as hotels. It also inspired our collaborations with Western, which we'll talk about in a second. Yes, so beyond our own team and community, our team collaborated with the cGEM team from Western University. We met with members from their human practices team to discuss microplastics. Their team was focused on policy development surrounding freshwater pollutants, including hormones and microplastics. Our main goal was one of scientific guidance, so providing some background on the basic principles of microplastics. For example, our team explained how microplastics are formed, the role that industry and water treatment plants play in the formation and collection of microplastics, as well as some of the methods that can be utilized to break down and recycle microplastics. Now, the knowledge of the microplastic filtration sub-team was the most applicable to what the Western team was trying to accomplish with their policy development. So understanding the processes that are involved with collection of microplastics, how the facilities are currently equipped to do this collection, and what happens to the collected microplastics all relates to the goal of this proposed policy document. Now, the human practices team from Western also joined our BioEnvo team during our Big BioGenie conference to present their industry-specific policy section on the prevention and improved regulation of microplastic use. Externally, we came across a PhD biology student from Carleton University named Jesse Vermeer, who conducted a study on Ottawa River microplastic concentrations both downstream and upstream from the wastewater treatment plants. Jesse provided us with his thesis and answered many questions we had regarding clarifier designs, accumulation of microplastics in sludge, and then further got us into contact with Professor Ormesi Banu from Carleton University in the Department of Civil and Environmental Engineering. Dr. Banu provided us with useful information about the nature of sewage sludge. She informed us that it is very difficult and very expensive to separate microplastics from sludge, and it would therefore be wiser to extract or remove microplastics by physical or chemical means before they are retained within the sludge. So she also suggested that the only true solution to prevent plastics from entering our waters is to stop producing single-use plastics in the first place and to focus rather on eco-friendly products. So this actually drove our team to come up with business solutions for recycling the extracted microplastics and or converting them into usable forms of energy by means of biodegradation. And on the topic of business, we've actually done some number crunching and looking at the cost of capital investment, the operating expenses, and potential revenue we can generate. For our business model, we would expect to get a return on investment in about 11 years, which may sound like a lot at first, but it's quite typical for projects of this nature. And then to scale the project, we would plan to expand our facility capacity to accommodate the microplastic waste produced by the 2 remaining wastewater treatment plants in the National Capital Region, so in Gatineau and Kemptville. This project will also yield significant environmental and social benefits for many stakeholders, including farmers, who will receive more purified sludge for their fields, the City of Ottawa, who will be recognized in a national and international context for environmental remediation of microplastics, and for our national economy, as it will provide a cost savings and sustainable alternative to crude oil during periods when crude oil prices rise particularly high. Now, when considering regulations for the project, it will likely require a variety of permits for lands, constructions, and use of various pieces of equipment in the process, as well as following guidelines from the Environmental Activity and Sector Registry when handling and transporting the plastic waste. Fundamentally, our project has 2 major target customers, so municipal wastewater treatment plants and industries that manage significant amounts of clothing material, such as hotels, laundromats, that kind of idea. For wastewater treatment plants, our objective would be to pitch the microplastic extraction system using ferrofluids, while we hope to pitch the laundry machine filter to industries. Ultimately, there are a number of problem constraints to consider when pursuing this endeavor. First and foremost, the implementation will be a significant challenge given the current lack of potential investors, funds, and customer interest. The market for pyrolyzed products also isn't presently developed. Another constraint is the logistics. There is no plan or consideration of how 22,000 plus kilograms of microplastics will be transported from the wastewater treatment plant to our facility every day. Given that we'll be working with the public sector, it is highly likely that a public procurement will also need to be issued. When looking at the local market context, currently there are no companies or markets that exist for collecting, sorting, and treating microplastics. The only potential competitors are plastic recycling and pyrolysis companies such as BBL Energy and Plasco, as shown in our chart. However, both those companies only accept larger pieces of plastic for their feedstock. We would be the only company operating in the microplastic processing space. Finally, to hear a little bit more about our business model and entrepreneurial communication, please check out the 5-minute pitch deck prepared by Kirsten and Vanessa for more information. And that's it. So a big thank you once more to Dr. Mads Kern for getting the ball rolling, to everyone involved in the BioGenie program for their efforts this summer, and to our external partners for sharing us with their time, their expertise. And if you have any questions, please don't hesitate to reach out. And thank you for listening to this presentation. We look forward to seeing you on conference day.